I'm really sorry, everybody. Peter unfortunately couldn't be here today. He's traveling for some work stuff, but I'm Brittany Kuhn. I'm a postdoc in Peter's lab, and I'm one of the ones who's really been highly involved in this UO1 at our site. And so today, you're pretty much just going to hear peer behavior from us today. Where hopefully this time next year we'll have some awesome genetic and epigenetic stuff. The genetic stuff is just rolling in, but we're going to stick with behavior today just to introduce our model to you guys. So some of this behavior is stuff that we are actively in the publication process with, and some of it is a publication we're hoping to kick out soon. Okay. If we want, there we go. Okay, so just like a lot of the other talks we've heard today, we naturally use a rather extensive behavioral protocol for our modeling. We put animals through 12-hour heroin self-administration training sessions. We then do a progressive ratio test about 3 weeks into that to look at motivation to continue to take infusion when cost increases. We do reinstatement tests, both a prime and acute test, and then just some extinction training in between. And bookmarking both ends of this heroin experience, we do other behavioral assays to allow us to be able to assess differences in anxiety-like and stress-like behaviors, as well as analgesic threshold. So the main goal of our model is we really wanted to be able to capture more of a syndrome perspective of addiction, as opposed to looking at how maybe one or two traits could map into addiction vulnerability versus resiliency. So in order to do that, we kind of looked through our behavioral paradigm and we selected 7 behaviors that we were interested in. And so our behaviors range from heroin reinforced versus heroin non-reinforced behavior, as indicated by the asterisk, but they also cover different, what we're saying, phases of the addiction process. So you can see we have certain behaviors that are representative in the consumatory or drug taking phase, others in the refraining phase, and then others in the reinstatement or relapse phase. And our objective here is we wanted to see how multiple traits across the entire addiction process interact with one another, conferring that vulnerability versus resiliency. And in order to do that with our collaborators at Ohio State University, we're using network-based clustering approach, which I can tell through several of the presentations so far you guys are all very comfortable with, so I won't really dig too deep into that. One thing I do want to mention, though, with our behavioral protocol, which is a little different than I think some of the others presented here so far, is that we run our animals in parallel with a sister site. So we run our animals here at MUSC in South Carolina, and then also at Dr. Roberto Chico-Chioppo's lab at University of Camerino in Italy. And our objective here with that is that we really wanted to be able to capture how differences in environment, may or may not affect behaviors or epigenetic factors that could contribute to heroin vulnerability versus resiliency. So naturally, though, when you run animals at 2 different sites, no matter how standardized you are, you're going to get differences between the 2 sites. And as you can see here, we very much capture that with MUSC having a bit more of a potentiated levels when you look at the raw data compared to Camerino. And I think this is a little bit more exemplified when you start to push our data into our network clustering analysis, where you start to see just each individual animal is 1 data point. So their behavior has not been reduced down to a solitary value. And we see MUSC, which is in pink, and Camerino, which is in blue, are rather distinct from one another. So before continuing with our analysis, we obviously had to standardize our data within each site. So we just did a Z-score transformation within each site prior to recombining the data. And when we do that and push, Z-scored transform data through the pipeline, we now see that our data between the 2 sites are really nicely interdispersed with one another, giving us the confidence to just go ahead and move forward with analyzing all this data together. And just so you guys know, the data you're going to see today is from about 450 of our rats. To date, we're up to about 750 rats. But you're just seeing where it was at a few months ago. right, so the next thing we had to do was really define, one of the main advantages of these clustering models is the fact that you are able to pull out these subpopulations or clusters that uniquely interact with one another and are related to one another. In order to do that, though, we did have to look at the Bayesian information criterion to really assess, based on the complexity of our data, our total N, the number of traits we put through, how many traits we actually are mathematically able to pull out of this analysis for it to be valid. So the BIC is just, your way of giving you the number of clusters that is the best fit for your data. And so we landed in between 3 to 5 clusters mathematically. And so then biologically, we, just had to kind of think about what made the most sense. And we decided to move forward with using 3 clusters. So that we could have a vulnerable group, a resilient group, and then just kind of this intermediate group. This is kind of our first pass through using this modeling technique. And as you can see, when we did that, we did get 3 really nice distinct clusters from one another, as to be expected. So we have our cluster 1, which is our vulnerable group, cluster 2, which is our intermediate, and then cluster 3, which is our resilient group. And I will show you their behavior in just a moment, so you can just understand why we termed all these guys vulnerable through resilient. Another check, though, that we wanted to do with using this modeling technique for behavior was to make sure that these models were, or these clusters rather, were in fact distinct from one another. It's very easy to look at this data and see, especially on the cluster borders, that we have some animals that kind of overlap a little bit. And so we wanted to make sure that we don't have an animal that's spending, 51% of the time in one cluster and 49% of the time in the other, especially because as we start to look at, the genetic and epigenetic profiles of these animals, we kind of just need a little more certainty in their placement in the clusters. So in order to do that, we just looked at an uncertainty measure to look at the cluster independence. And so again, the color coding is just matching with which cluster. So cluster 1 is vulnerable, cluster 3 is resilient. And the size of the dot is giving you an indication of its uncertainty level. And essentially the take-home message here is, except for 16 out of 455 rats are showing a little bit of uncertainty, but a vast majority of these rats are spending more than 90% of their time in one cluster. So they're pretty well classified as belonging to that cluster. If we look now at the Z-score transform data of the differences between clusters, so I think a good amount of it is kind of what you'd expect, in the sense that, our vulnerable cluster is showing, higher escalation, more consumption, motivation, having a harder time refraining, and then, greater relapse or reinstatement rates. But what we really want to emphasize with this data, and I think, what we all do using HS rats and our different modeling, is just the behavioral heterogeneity that we see within each cluster. So you do see that we have a good amount of overlap between animals, for example, in consumption, that are up here, even up here, but they're not in cluster 1, they're in these other clusters. And, with these violin plots, it's with the shape of the violin, you can see this, but we've also resorted to showing people this relationship just using just traces. And again, what we just really want to emphasize here is that we're capturing behavioral heterogeneity that we hope to capture, because we want to capture what we see in the human population. And so just because, for example, an animal in our vulnerable cluster, high escalation or high consumption does not necessarily confer high relapse rates. We just highlighted a few traces in each to give you an idea of some of the different behavioral profiles between these clusters. So it's really kind of indicating to us that by using this network clustering approach, we're able to see now how multiple traits are interacting, and it's giving us a better idea of, of classification of vulnerability versus resiliency. So it's not just based on one particular phase of addiction here. One thing, though, we did have to consider is we do have, males and females in here, which I think all of us are using. And so naturally, we did find a pretty massive sex effect with females outperforming males in virtually every single task, except for the last day of extinction training. So if we also look at how that maps into cluster composition, Not too surprisingly, we see, if we just focus more on this graph over here to the right, that percent of cluster, there's more females being represented in our vulnerable cluster as opposed to our resilient cluster. So what we've already started to do, and is part of the publication we're going to be pushing out soon, is we separate now males and females in the clustering analysis. Oh, I'm sorry. Just to be able now to get more of an understanding if there's a difference in the addiction profile between the two sexes. What time? I have a minute left. Okay. So what we've done with a subgroup of animals in MUSC moving forward is we wanted to look at punishment-based training. And so what we did is, after all of our training was done, we allowed a subset of animals to reacquire heroin self-administration training. So, everything as normal. And then by day 3, they pretty much all hit their baseline responding where they were during training. And then we put them through punishment-based training, and they had a 50% probability of receiving a foot shock with an earned infusion. And we did that over the course of 4 days. These are still 12-hour sessions though, so I think we hit a little bit of a bottom out effect here. I think we just hit them a little too hard. So I'm just going to show you data for this very first day where we used 0.4 milliamp shocks. And if you separate rats into their clusters, we do see that cluster 1 does show greater reacquisition compared to cluster 3. So presumably after a relapse episode, our vulnerable group is acquiring much more. And then if we look at our punishment-based training, we do see that our cluster 1 still continues on the whole to take heroin. We see a decrease also as well in our cluster 3 group. Although to be fair, most of them were pretty low to begin with. So most of them pretty much stayed even with where they were. And if we separate it into males and females, again, we see a sex effect with our females still showing greater taking behavior during this punishment task. But again, kind of the same trend where our animals in cluster 1 are continuing to take a little bit more than our cluster 3 animals. All right, so I know I'm about 12 and a half in. So, just some quick conclusions. We're super excited about this network clustering approach, and we're definitely going to be continuing down that road. I didn't have time to talk about HRLR stuff. I know that a lot of people in this group are interested in novelty-induced locomotor behavior, but we're going to have to save that, I guess, for a different talk. And we do show that kind of akin to what we see in the human population, females are showing more vulnerability compared to males in certain traits associated with heroin addiction. Moving forward, and hopefully next go around at next retreat, we'll have this data. Abe's already been processing some of our samples, and we have some really cool stuff that we're hoping gets maintained. We're also shooting off brain and blood tissue samples to Gary Hardiman at Queen's College in Belfast, and Kuhn Lab, no relation, up in Detroit to look at gut microbiome. We've also been doing at our site, we have some preliminary data now about some really cool differences in accumbens neuroplasticity between the clusters. And so that's something exciting we're continuing with. And also just now looking at how certain behavioral antecedents could possibly affect clustering assignment. So specifically looking at how acute stressors could possibly push animals from one cluster to another. I don't really have time to look at this, so just pretend like you didn't see that. But yeah, just big thank you overall to everybody involved. And these are our epigeneticists, Abe's group, the Ohio group, which is our statisticians, Leah and her crew, and then we have the 2 behavioralist groups. All right, I have that I did 14 minutes and 40 seconds. So I think I fit. That's about what I have.